is Cassandra Mapano, a senior at Tesoro High School.
Hi, my name is Anya Amin, and I'm a senior at Valencia High School.
My name is Yuki Matsumura, a sophomore at Woodbridge High School.
According to the CDC, 1 in 10 pregnant women report alcohol use.
And our project is titled Investigating the Effects of Alcohol on Embryonic Abnormalities. The project aims to answer to the question, how does alcohol affect embryogenesis and lead to congenital defects? We first conducted background research on the challenges with fetal alcohol syndrome disorders, as well as the role of ethanol metabolism in shrinking embryos. And we came to the hypothesis that if the chicken and duck embryos are exposed to this ethanol and PBS solution, then they will have lower survivability rates based on their reduced eye diameter growth, slower heart rates, and reduced spinal cord development. Some of the equipment that we used in this experiment include an ESNY dye to better depict the embryos, a heating gun, 36 Rhode Island chicken eggs and 30 Pekin duck eggs, a microscope for egg imaging, tweezers for egg windowing, an incubator to store the eggs, as well as a syringe, the solution itself, tape, markers, and a carton.
In this experiment, we analyzed 8 chicken and 5 duck eggs from the AA laboratory in San Diego, which were windowed after day 3 of incubation. There were 4 control and 4 ethanol eggs for the chicken and 2 control and 3 ethanol eggs for the duck. The eggs were windowed using the sharp end of a tweezer. We created a 70 milliliter mixture of 50% ethanol and 50% PBS solution and introduced 70 microliters of the solution into our experimental eggs. And then for the control group, we introduced only 70 microliters of PBS solution. After about 22 hours of incubation and introducing our dye to it, or stain to it, we observed these eggs under the microscope. Our tested variables include the eye diameter, the heartbeat, and the spine length. As for our results, the survivability rate for the control group for duck was 67%, whereas for the ethanol group for duck was only 25%. And as for the chicken, the survivability rate for the control group was 50%, whereas for the ethanol group, it was only 20%. So this shows a clear correlation that the eggs that were introduced to ethanol had a higher morbidity rate. Additionally, we noticed that there were a lot of irregular heartbeats. Some embryos had an increased heart rate, some had a decreased heart rate, and some had a complete heart block.
Based on our results, ethanol-exposed chickens and duck embryos exhibited a significant decrease in eye diameter, chickens being negative 0.07 milliliters and ducks negative 0.31 milliliters, while their spine length, chickens were negative 0.50 milliliters and ducks were negative 0.29 milliliters, compared to the control embryos, confirming that ethanol-induced growth retardation based on P is less than 0.05 statistic. The chicken eye diameter increased by 0.07 when exposed to ethanol, unlike the average shrinking observed in both chicken and ducks, with one outlier being Dongo, displaying a notable increase of 0.20 millimeters, potentially due to the human error during ethanol exposure or eye diameter measurement. However, overall data still indicated a trend of ethanol-induced reduction in eye diameter and spine length compared to controls. Our study on the effects of ethanol on chicken embryos and duck embryos aims to address the stigma associated with fetal alcohol syndrome by raising awareness, educating communities, and challenging misconceptions and stereotypes. By fostering inclusivity and promoting early interventions, we strive to reduce discriminatory behaviors and improve the quality of life for individuals affected by FAS and their caregivers, ultimately facilitating their access to resources and support.